Ever wondered how scientists decode the mysteries of life, unraveling the secrets hidden in our genes? Welcome to the intriguing world of bioinformatics, where the digital and the biological worlds collide. Bioinformatics is a rapidly growing field that leverages the power of computational tools to make sense of biological data. In the past few decades, this field has become an essential part of modern science, helping researchers to decode the secrets of life encrypted in our genes. The data that bioinformaticians often deal with are sequences of DNA, RNA, and proteins. Think of DNA as the blueprint of life, RNA as the messengers within our cells, and proteins as the workers that carry out all the functions. These sequences are like complex languages, and understanding them can give us insight into the fundamental processes of life. Unraveling these sequences is like deciphering a complex language, and that's where bioinformatics comes in. Imagine having an instruction manual that guides the development and functioning of a living organism. That's essentially what DNA is. DNA, or deoxyribonucleic acid, is one of the fundamental data types in bioinformatics. It's a long, twisted ladder-like structure, a double helix, made up of 4 types of molecules called nucleotides: adenine, thymine, cytosine, and guanine. These nucleotides pair up in a specific way to form the rungs of the ladder, creating a unique sequence that carries the blueprint of life. This blueprint harbors the instructions for the creation, development, and functioning of all living organisms. From the tiniest bacteria to the towering sequoia trees, from the humble housefly to us humans. But how do we read this intricate code? How do we access this vast library of information? That's where platforms like next-generation sequencing come in. These high-throughput technologies allow us to acquire DNA sequences from a variety of samples at an unprecedented scale and speed. They read the DNA, turning the physical structure into a digital sequence of A's, T's, C's, and G's. This sequence, this string of letters, becomes data that scientists can analyze and interpret. Bioinformaticians, in particular, play a crucial role in this process. They use advanced computational tools and algorithms to analyze these DNA sequences, to understand the instructions hidden within. They search for patterns, for variations, for anomalies. They compare sequences from different organisms or different individuals to uncover the mysteries of evolution, heredity, and disease. It's like being a detective, sifting through clues, piecing together a story from fragments of evidence. Each DNA sequence is a unique puzzle, a unique story. It may tell the tale of how a simple cell develops into a complex organism. It may reveal the secret workings of a deadly virus, or it may hold the key to curing a genetic disease. Each strand of DNA is a unique story waiting to be read, and bioinformatics provides the tools to interpret it. And if DNA is the blueprint, then RNA is the messenger, carrying those instructions to the rest of the cell. RNA, or ribonucleic acid, is another fundamental data type in the world of bioinformatics. It's like a faithful courier, diligently delivering the DNA's instructions to the protein factories within the cell. In this way, RNA plays an essential role in the synthesis of proteins, the workhorses of the cell. Now you might be wondering, how does RNA carry these instructions? Well, it's a process known as transcription. During transcription, a segment of DNA is copied into RNA. This RNA sequence then serves as a template for the production of proteins. This process is not just fascinating, but also holds the key to understanding how cells function, how diseases develop, and how potential treatments could work. And that's why bioinformaticians are incredibly interested in RNA sequences. To acquire this RNA data, scientists use a technique known as RNA sequencing, or RNA-seq for short. RNA-seq allows them to capture a snapshot of the RNA sequences present in a cell at a specific moment. This information can provide unique insights into the genes that are active or expressed at that time. By analyzing these RNA sequences, bioinformaticians can understand the genetic instructions that are being acted upon. They can identify what proteins a cell is making, how much it's making, and when it's making them. This can help them to unravel the complex mechanisms underlying health and disease. Moreover, RNA sequences can also shed light on different types of RNA molecules. such as messenger RNAs that carry the genetic instructions from DNA to the protein-making machinery, and non-coding RNAs that regulate gene expression. In essence, RNA sequences provide a dynamic view of the cell's activities in real time. They act as a bridge connecting the static world of DNA blueprints to the dynamic world of proteins. So if DNA is the master architect, RNA is the project manager, ensuring that the blueprints are transformed into reality. And as we delve deeper into the world of bioinformatics, we come to realize the immense value of these messengers of the cell. RNA sequences act as the bridge between the DNA's blueprints and the proteins that carry out the cell's functions. With RNA carrying the DNA's instructions, it's the protein sequences that put those instructions into action. Imagine protein sequences as the diligent workers of the cell, taking the messages from RNA and turning them into tangible action. Proteins are the end product of the genetic code, and they perform an astounding array of vital functions within the cell, from building structures to fighting off invaders. These sequences, then, are a crucial data type in the field of bioinformatics. Protein sequences are translated from RNA, a process that's like decoding a secret language. Each three-letter sequence of RNA, known as a codon, corresponds to a specific amino acid. When these amino acids link up in a chain, they form a protein. And this isn't just a random string of amino acids. Oh no, each protein has a unique sequence, a specific function, and a custom fit within the cell's intricate machinery. It's like a tailor-made suit designed for a very particular job. But how do bioinformaticians get their hands on this protein sequence data? Well, one popular method is through something called mass spectrometry. This technique allows scientists to identify the unique mass of each amino acid in a protein, and from there they can determine the protein sequence. It's like piecing together a complex puzzle, each piece revealing a little more about the overall picture. This data is invaluable for bioinformaticians. By analyzing protein sequences, they can predict a protein's function, understand how it interacts with other molecules, and even unravel the mysteries of diseases at the molecular level. It's like having a detailed map of the cell, guiding them through the labyrinth of life's smallest units. So, you see, protein sequences aren't just strings of amino acids, they're the workers of the cell, taking the instructions from RNA and turning them into action. They're the tangible expressions of the genetic code, the hands that build and maintain the cell, the soldiers that defend it. Protein sequences are the end product of the genetic code and the workhorses of the cell, performing tasks based on the instructions encoded in DNA. From the blueprint of DNA, to the messenger RNA, to the worker proteins, each sequence plays a crucial role in the grand scheme of life. These sequences form the basic data types that bioinformatics tirelessly seeks to understand. DNA sequences, the long, twisting ladders of life, encode our genetic information. RNA sequences, the tireless messengers, take this information and transmit it throughout the cell. And the proteins, they're the workers, the doers, putting genetic instructions into action. These sequences are interconnected, each one a crucial part of a complex, beautiful dance that is life itself. Bioinformatics is the tool we use to understand this dance, to decode the intricate patterns and rhythms of life. It's through bioinformatics that we can see the connections, the interplay between DNA, RNA, and proteins. Bioinformatics, in essence, is the key to deciphering the language of life, one sequence at a time.